Today's class is on ocean acidification. And by the end of this class, you guys should be able to understand what ocean acidification actually is, and also describe the ways that ocean acidification affects species. Now, one of the reasons I want to talk about ocean acidification specifically is that between the years of about 2013 and 2016, a large part of my own personal research program was specifically looking at ocean acidification. This was a large part of my life during that time, and I thought it would be worth dedicating some class time to it. And in this class, we will actually talk very specifically about some of the work I did during that time. Just before we get started, I thought we'd have a think about, well, what do I know about ocean acidification? So think about this. You could select A, I've never heard of it. B, I've heard the phrase, but I don't really know what it means. C, I've heard the phrase and understand that it could be bad. D, I understand the issue, but I don't know how it works. Or E, understand the issue, the process, and the chemical reactions. Just take a couple of seconds to think about where you fit on that scale. Okay, well, the reason I brought up ocean acidification is because in the marine world it's becoming more and more serious, and it's actually been labelled the other CO2 problem. When we think about the issues with CO2 getting released in the atmosphere, we might think about temperature, but ocean acidification has been defined as the other CO2 problem, one that was percolating away this whole time, but we didn't really pay that much attention to. Because in class previously, we discussed and looked at the relationship between CO2 and temperature. So in this figure here, on the x-axis, we have thousands of years ago. So this is the present day. This is going back thousands of years. We have temperature. Remember, this is taken from the ice cores. So temperature has been changing. And the green is CO2 in the atmosphere. And you can see this has been changing about the same rate as temperature has. In fact, it's not about the same rate, it's almost exactly the same rate. The lines line up really, really closely, the peaks and troughs. Okay? We also on this one have dust, but we're not really going to talk about that. Now this CO2 gets released into the atmosphere and it does change temperature. However, one thing we need to think about is that about 26% of released CO2 is actually absorbed by the oceans. So our activities such as burning of fossil fuels and deforestation is releasing this CO2 into the atmosphere, and about a quarter of that CO2 is just absorbed directly by the ocean. And in the ocean, this CO2 combines with water to produce acidic compounds according to the reaction shown below. So let's start at the far side. Here we've got carbon dioxide. This is carbon dioxide in the atmosphere. When it's absorbed into the ocean, it combines with the H2O. So H2O, remember, dihydrogen oxide is water. So this is the seawater. It's absorbing carbon dioxide from the atmosphere. CO2 combines with H2O and it produces this compound called carbonic acid. This is the first compound that's created when CO2 combines with H2O. However, this compound then dissociates further and we get free hydrogen ions and bicarbonate ions being produced. These free hydrogen ions are what then makes the ocean acidic. They're what lowers ocean pH. And these acidic compounds, when they get into the ocean, do begin to actually lower pH. And we can actually measure this using the Mauna Loa station and the Aloha seawater stations that are both present in Hawaii. We talked previously about the Mauna Loa atmospheric CO2 station. The reason that the global CO2 measurement is taken from Mauna Loa is because it's in the middle of the Pacific Ocean, so it's seen almost as like a base level of CO2. What we have here is, on the x-axis we've got year, and on this y-axis we've got CO2. This is what we'd seen before. We'd seen the CO2 in the atmosphere at Mauna Loa increasing. Now we can also look at the CO2 level in the ocean because we've got the Aloha seawater PCO2 in situ. This is the CO2 increasing in the ocean over the same period. Unfortunately, we only started collecting these data in the late 80s. Also, we can see what's happened to the pH of the ocean at the same time. And the pH of the ocean, so the acidity of the ocean, has increased, which has led to a decrease in pH between the years 1988, and this one only goes as far as 2011. So what we can see is we're already measuring increased CO2 in the atmosphere, leading to increased CO2 in the ocean, leading to decreased pH or increased acidity. But these recorded changes in pH seem very small. I mean, should we really worry about it? Well, the pH change that we've recorded in the ocean, not that we're predicting, but that we've actually recorded, that pH has changed from 18.15 to 18.05. So a drop of 0.1 for pH point. Well, the first question is this a lot? It doesn't sound like an awful lot, but so what is it an awful lot and what sort of effect is it having? Well, pH in the oceans is historically very stable. The oceans themselves have a buffering capacity that means that the ocean pH doesn't really change at all. In addition, many ocean species have skeletons made of calcium carbonate. So all the oysters, all the crabs and lobsters, all the sea urchins, all the abalone, all the nautilus, squid and cuttlefish, and all the corals, their skeleton, so the hard structures within inside these species, is made of calcium carbonate. This is what generates limestone. And these calcium carbonate skeletons dissolve very, very easily when conditions become slightly more acidic. So this is an image that has essentially become synonymous with all of ocean acidification research. It shows the shell of a gastropod. The gastropod was raised in regular ordinary seawater until it was this size. When it got to this stage, a whole load of these gastropods were then placed into acidified ocean seawater. So this is ocean seawater at the pH we expect it to be in the year 2100. And they were left there, and what we can see is between day 1 and day 45, the shells of these snails started to degrade, break down, until eventually they were almost unrecognizable. So what this means is the skeletons that are present in every crab, every lobster, every coral, every oyster, every mussel, every sea urchin, every starfish, and every single oyster and every single clam, all of these skeletons are made of this calcium carbonate, and they have the potential to dissolve or not grow properly if we increase acidity or decrease pH. And laboratory experiments that have been conducted recently have revealed the effects of ocean acidification on individual animals. So in these images here, this sea urchin is grown in just ambient seawater that's floating around in the oceans today. This sea urchin, which is actually the sibling of this sea urchin, both of these they're essentially brothers. This sea urchin was grown instead in seawater where the pH conditions were that we would expect in the year 2100. And what we see, for example, is these pencil urchins, they lose their spines, so you can see, look, hardly any spines compared to this fella, and they're reduced in size. They shrink in size generally. So both these urchins were taken as tiny little larvae, a couple of millimeters long, both placed into water that is in the conditions it is today, and water that's conditions that are predicted for the year 2100, grown, and we see huge changes in their spines and also a decrease in their overall size. In addition, when we look at coral, so these are coral that are grown in a lab from different oceanic conditions. The ones with yellow tape are in ocean conditions like they are today. The ones without yellow tape are under CO2 conditions we expect for the year 2100. And what we see is when we've got, in current conditions, the coral is very branched, it's very healthy, we've got lots and lots of polyps, it's branched left, right and centre. When the CO2 conditions are high, which has led to increased acidity. We've got much less branching. The coral is not nearly so clustered, and we've got far fewer polyps on the end, and this is seen again and again. So it looks like for both sea urchins and coral, bearing in mind these two species are very unrelated, both of them show serious changes in morphology when they're placed in high CO2 conditions. So just to check up on the clinical question, just think this to yourself. So why is a small change in pH such a problem? A, the oceans have historically been very stable in terms of pH. B, many marine species have calcium carbonate skeletons that easily dissolve. C, both A and B. D, species get burned by the acidic oceans. E, reduced pH will reduce oxygen levels. Just give you 10 seconds to think about that. 5, 4, 3, 2, 1. Well, in this case, the answer is A. The reason that oceanification is a small change is such a problem is because the oceans have been historically very stable in terms of pH, and many marine species also have calcium carbonate skeletons that easily dissolve. And these two factors combine to mean that even a tiny change in pH is a real problem in the ocean. Okay, so these lab experiments showed what happens to individuals when they're placed in ambient or high CO2 conditions. But the question then is, what determines which individuals are present in a community? We can look at the effect of CO2, high CO2 on individuals, but what about whole communities? Well, remember, a community of animals is made up of a lot of populations of different animals. And for each population, that population is made up of a collection of individuals. So what we're asking is, we've done these experiments on individual animals, but how does that scale up to affect the structure of entire communities? And first, we have to ask ourselves, well, what determines which individuals are present in a community? Sir John Lawton, who was a British professor, popularised the filters concept that determines who's present in the community in 2000. We actually visited this a while ago. So what we do is, we have the filters concept. So right at the top, we have the regional species pool. This is all of the species that exist in the area that we're interested in. And what we're trying to do is work out who actually survives in the local community. So, here's our starting point, our regional species pool. First of all, each individual species has to pass through the dispersal or immigration filter. And this filter weeds out species that are unable to reach a specific habitat, i.e. they can't swim, fly, disperse seeds, or something like that. And what we're left with after we pass through the dispersal filter is we're left with the group of species that are able to reach our focal location. From this pool of species, each individual species then has to pass through the abiotic filter. And what this abiotic filter does is this filter weeds out any arriving species that is unable to cope with the new environment, i.e. it can't cope with the pH, it can't cope with the temperature. For example, it's either too hot or too acidic. Then we have the species that pass through this abiotic filter, and this is this group of species that are able to make it. To get to the final local community, these species then have to pass through the biotic filter, which is based on species interactions. And this biotic filter weeds out species that are unable to compete or deal with predators, or they're lacking prey or pollinator species. So what we have is, we start off with our regional species pool. Each of these species, to get to the local community, has to pass through the dispersal filter. Then it has to pass through the abiotic filter. Then it has to pass through the biotic filter. And then we end up with our local community. So let's think about this in terms of ocean acidification. So we have the determinants of a local community, again our ecological filters concept. Here, for example, is our potential community. This is the species and the various abundances that could exist in our regional pool. What we find is the abiotic filter removes individuals in our community that are incapable of dealing with local physical conditions. After the abiotic filter, we then have the biotic filter, and this then removes individuals incapable of coexisting with other local community members. From this, our local community remains. So this is just applying the basic filters concept to a community of marine organisms. We can then ask the question, how may ocean acidification, or as we shorten it OA, affect the strength of these filters and therefore affect overall community composition? Well, remember, ocean acidification affects invertebrate physiology. So we've shown before how these gastropods have these changes in shells. And this change in physiology happens especially for species with hard exoskeletons. But these skeletons help protect against predators. The skeletons that are present on coral, snails, crabs, lobsters, sea urchins, they're not just there for show, they have a functional role. And crucially, these skeletons help protect the organism that has them against being eaten by predators. Crucially, jellyfish that are present in the ocean appear relatively tolerant of ocean acidification. They don't seem to get affected by ocean acidification really at all. And crucially, jellyfish are predators of plankton. Plankton are the small, tiny animals that float around in the oceans and provide food for pretty much the entire rest of the food chain. And jellyfish eat plankton a lot. Jellyfish are also soft-bodied. They have no hard exoskeleton. And previous experiments on individual jellyfish have shown that these species can survive and reproduce even at pH as low as 7.2. And remember, the current pH in the ocean is 8.05, and that's down from 8.15. So these jellyfish can survive at pHs much, much lower than what we're seeing now. In other words, they can tolerate very acidic conditions. So I asked the question, does this disparity in the effects of ocean acidification make prey more susceptible to predation? In other words, how do zooplankton prey, which possess these hard calcium carbonate exoskeletons, how do they deal with the threat of predation under ocean acidification conditions? In other words, how does this ocean acidification affect the biotic filter? So what I did was I generated the following hypothesis. My hypothesis was that ocean acidification alters the strength of multiple filters. So if we start over here with our regional community, this has to pass through the abiotic filter. And ocean acidification changes chemical conditions. This should directly change the abiotic filter. Then we have our community of species able to pass through the abiotic filter. They then have to get through the biotic filter, which in the case of this experiment is the threat of predation from a jellyfish. But ocean acidification could indirectly change the biotic filter because it changes the shape of the skeletons that are present in prey, and this makes them more susceptible to being eaten by predators. So what I did was I designed an experiment where predator presence-absence was crossed with ambient or ocean acidification conditions. So what we have is this sort of 2x2 factorial design. So we have experimental replicates where we have no predator, and experimental replicates where we have a predator, in this case a jellyfish. We also have experimental replicates where we have ambient pH, which in the case of this experiment was conducted in Sydney. The pH of the ocean in Sydney, Australia is about 8.2. And also we have conditions where the pH was acidified and dropped to 7.8. So these are the danger acid versions. And what I did was I produced this schematic of different experimental conditions, and then I used a zooplankton community that consisted of copepods, plicosomatids, which are crabs, basically, dendrobrachiatids, which are a type of shrimp, amphipods, and mysids. So we had an entire zooplankton community, and I placed them into tanks in the laboratory, and each tank either had a no predator or a predator, and it also had ambient conditions or high CO2 conditions. What I then did was measured the abundance of different species within the communities in these experimental vessels. And what I found was, for some species, in this case calanoid copepods, a combination of ocean acidification and predators reduces calanoids much more than predicted. So on each of these figures is going to take the same form. On the x-axis, we have the experimental condition. And on the y-axis, we have the log abundance of different species. So this bar here represents a pH of 8.2, so essentially ambient CO2 conditions, and there's no predators. And we see an ambient CO2 and no predators, we get a lot of copepods. When we drop the pH or increase ocean acidification, we see a drop in the number of calanoid copepods, so they drop down from here to here. When we keep ocean acidification conditions the same as ambient, but we add a predator, we see that the number of copepods drops from here to here. But crucially, when we add both a predator and we change the CO2 conditions so that we get increased acidity, we now see that the number of copepods drops from here all the way down to here. And we can compare this to what we call a null model. And a null model tells us what we'd expect the population of copepods to be when we add ocean acidification and predators, using the results if we just drop the pH. So this is increased acidity. This is adding a predator. This bar here tells us where we'd expect the copepods to be if these two effects were just additive. So if this and this were operating independently, we would expect the abundance of copepods to be here. But what we actually see is in the situation where we've got both predators and ocean acidification, the number of copepods drops much lower than what we'd expect. What this means is the threat of predators and the threat from increased ocean acidification is acting antagonistically. It means that not only does ocean acidification directly negatively impact some of these copepods, but it also increases the chance they can get eaten by predators, which is why they're dropping so much. We actually found that the combination of oceanification and predators reduces plicomata or crab larvae more than predicted. This is a little baby crab. They're actually kind of cute. What we see is this is the amount of crab larvae you observe under ambient conditions with no predators. If you increase acidity, you actually get no real change in the number of crab larvae. If you just add a predator, you get no real change in the abundance of crab larvae. If you add acidity and a predator, the crab larvae actually drop down, which is much more than we'd expect based on a null model. Oh, just as a reference, these letters above the different bars, different letters above a bar denote bars that are significantly different from each other. So for example, all of these bars have an A, that means they're not significantly different from each other. This bar has a B, which means it's significantly different from all of these ones. Okay. And what we found when we actually looked at the direct impact of predation, we found that ocean acidification increases predation rate on both calanoid copepods and Plysiomata. So on this bars here, these two bars relate to calanoid copepods. These two bars relate to Plycomata, the baby crabs. The white bar is under ambient conditions, that's low CO2, low acidity. The grey bar is under future conditions, that's high CO2, high acidity. And what we can see is under current ambient conditions, the number of copepods that actually get eaten by jellyfish is slightly less than 40% on average. However, when we increase the acidity, the number of copepods that gets eaten more than doubles to over 80%. And under ambient conditions, crab larvae don't really seem to get eaten by predators at all, but suddenly when you add in acidified oceans, they get eaten an awful lot. Over 70% of the crab larvae get eaten. What this means is ocean acidification is increasing the ability of jellyfish to capture and eat their prey. We also found out that ocean acidification eliminates copepod nauplii. These are baby copepods. So in this case, when we've got ambient conditions and no predator, we have lots of baby copepods. Under acidified conditions, we get none at all. Even when we add a predator, we don't actually get many fewer baby copepods than under ambient conditions, but as predicted, a predator and acidified conditions just drops them down to nothing. What this means is that in terms of the entire ecological community, the community level effects of ocean acidification are realised through multiple mechanisms. So remember, we start out with our regional species pool, and it turns out from our experiments we can see that ocean acidification directly affects the strength of the abiotic filter because copepod and nauplii populations were reduced directly just by increased acidity. However, what we also found was that ocean acidification can increase the strength of the biotic filter. What happened was ocean acidification indirectly affects the biotic filter by increasing the predation on copepods and Pleisiomata. So what this means is at the community level, not at the level of just a single individual, but for whole ecological communities, the effects of ocean acidification are realised through multiple mechanisms. Critically, what this means is that the direct effects of ocean acidification do not need to be fatal in order to cause mortality. In other words, ocean acidification does not need to directly kill organisms in order to cause mortality, because it can just increase the chance that organism is eaten by its predators. It doesn't matter how you die, it's just that you die. So this research that I talked about, I actually published a couple of years ago in a journal called Global Change Biology. The paper is available. The title of it is Ocean Acidification Alters Zooplankton Communities and Increases Top-Down Pressure of a Cubozoan Predator. This was quite an important finding because zooplankton in the ocean are one of the biggest pools of biomass in the world, and so zooplankton in the ocean represent one of the world's biggest food webs, so it has quite important ramifications. Because of this, this research was picked up by the New Scientist. They did an interview with me and actually did a popular piece on this called Box Jellyfish Will Destroy Future Oceans by Gobbling Up the Food. It was a little bit sensationalist, but it pretty much got the idea across. And they also made it into the Guardian newspaper in an article entitled What Do Jellyfish Teach Us About Climate Change? This was the first time I was really featured and interviewed in the Guardian, and also the first time I ever had a photo published in the Guardian, which was kind of cool. So if you want to read more about this, sometimes these popular science articles are often the way to go. So, just a quick question to make sure people are still keeping up. So what were the consequences of ocean acidification at the community level? A. Ocean acidification reduced the densities of all species. B. Ocean acidification increased predation rates on the most abundant species. C. Ocean acidification negatively affected jellyfish health. Or D. Ocean acidification reduced predation rates on plankton. Just give you 10 seconds to go through that. Think about it. Five, 4, 3, 2, 1. Well, what I found was ocean acidification increased predation rates on the most abundant species. Okay, well, these experiments that I've talked about so far were all done in controlled laboratory conditions. So the experiments looking at the effects of oceanification on individuals, such as coral and sea urchins, they were done on individual animals, grown in a lab, and then watched through time. In addition, the experiments that I did at the community level were under highly controlled conditions. I set up a bunch of tanks in a lab. In each one of those tanks, I put some zooplankton, and I put a predator, or I didn't, or I changed the pH conditions, or I didn't, and then watch what happened. They're in very controlled conditions. And so these lab experiments may not properly reflect conditions in the real world. So then the question starts to be, can we find real-world evidence of the impacts of ocean acidification? And the fact is, yes, we can. There are certain places in the world where we can go into the field and actually find real-world evidence of communities and ecosystems changing as a result of ocean acidification. Two of the most useful ways we can do that is shown below. So we can take repeated measures through time of the same parts of the world. This just happens to have been done coincidentally in certain areas, and we'll talk about one of those in a sec. So this allows us to measure changes through time in a single area, but we also have this fabulous system called volcanic CO2 seeps. These allow us to look at a very local scale how changes to ocean CO2 conditions affect the health of entire ecosystems. And we'll go through both of those now. So first of all, continuous monitoring is already documenting changes due to ocean acidification. So one of the key places this has been done is the Keys of Florida. Because due to their easy access, scientists have continuously monitored corals in Florida. And in the Keys, corals can no longer replace the calcium carbonate they lose in the winter months. Essentially, the corals in the Keys are wasting away. And this has been photographically documented through time. So these two pictures over here on the left represent images taken at the same place in the Florida Keys at different times. So this image was taken in 1976. This was when ocean acidification, CO2 in the oceans was much lower than they are today. What you can see is this area is absolutely full of this branching stag's horn coral. This image here was taken in the exact same location 40 years later. And you can see there's literally no living coral left. So all of this branching coral has died and wasted away. We can even see the fragments, the remnants, the skeletons of these corals on the ocean bed. So this gives us an example of where we've taken continuous photos through time in the same location, and we've seen huge changes in the community of corals. Another way that we've monitored through time is the Continuous Plankton Recorder project. And this has monitored the world's oceans for 86 years. The way this project works is that there are little boxes placed on the underside of an awful lot of seagoing vessels, such as cruise ships, container vessels, and research boats. And what these do is they take, as the boat is travelling, they take a sample of the plankton automatically out of the water. They preserve that plankton sample. They then store it on the boat. When that boat returns to a nearby port, all of those samples are then shipped to the headquarters of the Continuous Plankton Recorder project, and then they're quantified, the abundance and distribution of different organisms is monitored. This project has actually been going for the last 80 plus years, and it's led to some incredible discoveries. One of the most astonishing ones was the story of the pteropod. So this is a pteropod. It's affectionately known as a sea butterfly. It's not very big. It's about 1/4 of an inch long. But these are essentially small planktonic snails. They're actually tiny snails that float around in the world's oceans. And due to their calcified shell, so this lump in the middle, this calcified shell is highly important to maintain their buoyancy. But due to their calcified shell, they're considered highly susceptible to ocean acidification. And populations are significantly reduced in many parts of that word should actually be world, not works, I apologise. So the Continuous Ocean Plankton Recorder project has documented that all over the world, populations of these pteropods are massively reduced and in some cases have entirely disappeared. And in addition to these continuous monitorings, we also have CO2 seeps. And CO2 seeps occur all over the world, and they allow us to estimate future ocean conditions. So here, for example, is an underwater image of carbon dioxide seeps in Papua New Guinea. This is awesome because Papua New Guinea is tropical, so we actually have what happens with CO2 seeps in tropical areas. Also, we have them in the Mediterranean, so CO2 seeps in Italy. So this is not a coral reef ecosystem. This is a temperate ecosystem which is dominated by seagrass. And CO2 seeps occur in volcanically active areas. The reason these CO2 seeps are there is because lava and volcanic activity just underneath the seabed is generating CO2. This CO2 escapes through the seabed and enters the seawater from underneath, which is what these bubbles are here. And this CO2 is released from into the water from the earth. This CO2 then increases acidity in the same way that increased atmospheric CO2 would increase acidity. And the critical thing about these CO2 seeps is that they're a very localized phenomenon. They're not very big at all. Many of these CO2 seeps occur on a scale of a couple of meters. As a result, we can compare what happens in these CO2 seeps with normal conditions that are often found only a few hundred or tens of meters away from the seep itself. And what happens is these CO2 seeps show reduced coral diversity, growth, cover and health. So this is taken from the Papua New Guinea CO2 seep. This is what the reef looks like literally 100 meters away from the CO2 seep under ambient conditions. What we can see is we've got a lot of these plate corals. We've got a lot of branching coral. This is coral that's very fast growing and is particularly susceptible to changes in ocean pH because it grows so fast and it's not particularly strong. When we literally point the camera in the other direction and take a picture with inside the CO2 seep as seen here, we see we've got literally 0 plate corals or branching corals at all. Right at the seep itself, there is no coral growth. As we move away from the CO2 seep, bearing in mind in this region the CO2 in the water is a bit higher than it is in ambient conditions, but it's not as high as it is right at the seep itself. Here we can see we do have these corals, but they're boulder corals. There's no branching corals at all, and no plate corals. And we can actually measure CO2 and pH as we move towards or away from the seep and compare what we find to predictions of future ocean conditions. So here we have a situation where we're starting away from a seep and moving towards a seep. So if we start a long way away from the seep, we measure the pH, it's about 8.5, which it is today. We then move slightly closer to the seep. We now find the pH is 7.95, which is what we're expecting the ocean pH to be in the year 2050. We can see we've lost a lot of the basic coral diversity, and we've lost an awful lot of the things that we consider sort of iconic species of coral reef. If we move even closer to the seep, the pH there drops to 7.8. This is what we're expecting in the year 2100. This is also the pH I used in my ocean acidification experiments. And we see here, right at the seep, we've got 0 coral right at the seep, a little bit of boulder coral a little bit away. What this means is we have very good predictions of future carbon dioxide concentrations and can therefore calculate future ocean conditions. In addition to going towards seeps and away from seeps, these CO2 seeps don't remain in the same place the whole time. And new seeps appear, and we can actually document the appearance of new CO2 seeps in the water. When new seeps appear, we can actually get before-after pics, which you see here. So this is the same location. Here's what this particular stretch of reef looked like before a CO2 seep appeared. Here's what it looked like afterwards. And we can see we've got this dramatic change from a coral-dominated ecosystem to one that's dominated by turf algae. And these CO2 seep experiments all reveal similar patterns. In each case, we observe reduced diversity, increased dominance of just a few species, replacement of coral or coralline algae with fast-growing turf algae, and generally everything gets worse. Okay, so we just have a final fake eye clicker question. What do I now think about ocean acidification? A, it's not going to affect anything. B, it may have an effect, but it's not important as the ocean is a long way away. C, it may have an effect, but the oceans are big, it's not a problem. D, it could be a problem. Or E, it's one of the biggest threats facing the world's biggest ecosystems. Just give you 5 seconds to reflect on that. 3, 2, 1, go. Righty, just as a last word on this, I was going to show a video in this as well. This is a video, a lot of it is actually taken on a place called Heron Island where I've worked, and this person here, Ove Goldberg, I've actually worked with him a little bit. They did a big popular TV show a couple of years ago looking about collapse of the oceans. I'm going to show this video live inside the video I'm showing here, but I'm also going to put a link to this video into the description of my video so that you can watch it directly in YouTube.
Hogelberg is a marine biologist who has spent his life studying corals. More and more, his focus has been on climate change and the impact it will have on the world's reefs. But before I see how climate change will affect them, Ove wants to show me what's at stake. What greets me under the surface of the water blows my mind. The Great Barrier Reef is one of the 7 natural wonders of the world, and I can see why. And when I look closer, I see that reefs are supporting all of this life, providing food, shelter, and places to hide from predators. I'm realizing how interconnected everything is. Though they make up less than 1% of the ocean floor, reefs support 1/4 of all marine species. If climate change will alter the reefs, is all this life under threat?
We're seeing a change in the chemistry of the ocean that has no parallel in 65 million years, if not 300 million years. That's the last time it changed this quickly.
Ove explains to me that we've pumped more carbon dioxide into the atmosphere than there's ever been in human history. Scientists knew that this added CO2 was warming the oceans along with the rest of the planet. But what they only recently discovered was that about 1/3 of it ends up in the ocean. There, it combines with seawater to create a dilute acid. And here's the truly shocking part. Today's ocean is already 30% more acidic than it was before the Industrial Revolution.
And here's the kicker. It takes 10,000 years or more to reverse the changes that we've made already. That's the size.
It's an astonishing number.
It's astonishing, isn't it?
Yeah.
You're looking at an experiment we've been running over the last 12 months, and this experiment has been exposing parts of coral reefs to warmer and more acidic oceans. In this tank, we've just changed the acidity levels, adding CO2 to the water. The scientists working on this project have discovered that although they look well, when you actually look at the process of which they build their skeletons, and they've almost got a form of osteoporosis.
Right.
Now that's important because if you have a storm come through, these corals are going to break more easily. And so when you look at this next tank, now exposed to 12 months of 4 degrees Celsius above where it is today. And what you see here is that most of the corals are dead. Any coral that survived is bleached. You can see that.
Dramatic, frankly. Ove tells me that coral is actually an animal, and that inside its tissue, there are tiny brown algal-like organisms that provide up to 90% of the coral's energy. When the water gets too warm, the coral expels these organisms, becoming a ghostly white. That's bleaching.
So we've changed the acidity, we've changed the temperature, but what happens when we change both?
Oh my God. This to me looks like death. Truly shocking. The stuff that we're not seeing is not that you didn't put it in here, it's that it's dissolved.
It's gone. It's gone. And
It's this.
Yep, it's sand. And that's the future we're on right now. Keep pumping CO2 into the atmosphere. This is what we're going to get.
What I see next makes me realize how close this threat is. Footage coming in from the northern sections of the Great Barrier Reef shows widespread bleaching already underway.
That is far worse than I thought it was.
In some regions, parts of the reef have already died and been taken over by the slime that Ove showed me in his future scenario tanks. One of the 7 natural wonders of the world is disappearing before our eyes, and with it, the support system for millions of sea creatures.
Up to this bleaching event, how much of the world's corals were lost?
In the Caribbean, it's very clear that we have lost probably 80% of corals since the 1970s.
Hold on. 80% of the corals in the Caribbean are already gone?
That's right. I think you can safely say that we've probably lost 40 to 50% of corals across the planet in the last 50 years.
Yeah.
Within 20 or 30 years' time, you don't have coral anymore.
If we just continue as we're going right now, coral reefs will cease to exist by mid-century of this century.
Yep. 35 years from now.
Okay, and as I said, I'm going to put that in the actual description of my